I'm Dr. Rebecca Fisher, and I am doing your lecture today about infectious diseases epidemiology. So I wanted to mention first just the history of infectious diseases, quite an interesting history worldwide. So we've had major infectious disease shaping history, and some of these you guys will have heard about. So smallpox, which of course now is eradicated, but caused more deaths, we think, than any infectious disease event in history. Malaria is still a public health problem. The Spanish flu or influenza pandemic of 1918, bubonic plague, cholera, AIDS, and typhus. And many of these are still diseases that we see in the world, including in the United States. So obviously we see influenza regularly. Malaria is not endemic in the United States. It was eradicated from the United States. We do have cases of the plague, including in Texas. Cholera still is a major disease worldwide. AIDS, of course, still killing people, although with new medications, we've reduced that burden quite a lot. And then typhus. And typhus is a disease that we also have here in Texas. So historically, the Spanish influenza pandemic was one of the major disease events in history, one of the few declared pandemics, killed mostly healthy young adults, which was troubling because influenza typically, even in present day, kills the very young and the very old more than the young, healthy individuals. So your age, 20s, 30s, 40s, typically are not who we see suffering from severe disease due to influenza. So we think that this affected about a third of the global population. It's unknown exactly how many people died. Estimates range from about 50 million to 100 million people. But given the time that this occurred, so the era that this occurred, and reporting and disease surveillance. Certainly we didn't have email and computer networks. We didn't have online reporting systems. So individuals that were remote or in rural areas may not have been counted, including individuals sometimes in whole countries and continents. So think of Latin America, South America, think of countries on the African continent and in East Asia, so predominantly in the tropical belt, lower income countries that don't have resources and often don't get counted, and that's still the case today. So the second epidemic that really sticks in people's mind is the plague. This is also a global problem and killed, it's estimated a fourth of the people that were succumbed to the Spanish influenza pandemic. This is caused by a bacteria called Yersinia pestis, and this is carried by a flea. It bites rodents. It can bite humans, so this is a zoonotic disease, and it still occurs today sporadically in the United States, usually in the sort of southwestern United States. So in addition to causing disease, disfiguring, and death, we also have major impacts of communicable diseases that relate to sort of social, economic, and cultural parts of our life and history. So slowing industrial development, urbanization, it affects wars, it affects economic productivity, and then culturally, we see disease depicted in a lot of artwork and plays and operas. So of course, we have weapons against infectious disease, so vaccinations, treatments, things like refrigeration and sanitation, clean water, chlorination. These all help us to reduce infectious disease occurrence. And in fact, these are in existence today because we wanted to save lives. So another big win for public health was controlling vectors, so controlling malaria. So as I mentioned, malaria was eradicated from the United States, primarily through vector control through spraying insecticides where mosquitoes were. So all along the Mississippi River used to be a huge belt for malaria in the United States, but we were able to control this. So there are other vector-borne diseases as well that can be pushed back, let's say, controlled somewhat with vector control measures. So the crude death rate due to infectious disease has declined over the years due to these measures that we developed. So some things to mark in history include the creation of health departments, as I mentioned, chlorination, decreasing diseases. So the last human transmission of plague. So now we see it actually only as a zoonotic disease, which is transmitted from the animals to humans by way of the flea instead of from humans to humans. So we invented or discovered penicillin, vaccinations, and then sort of compulsory vaccinations. So this is the, on the x-axis year from 1900 to about 2000. So this is the crude death rate. So how many people per every 100,000 group in the population die. So we see this massive peak here. So this is a peak in deaths. So and this, you can see the impact of the influenza pandemic on the global death rate over the years. All right, so in infectious disease epidemiology, we're really trying to study infectious diseases in order to understand how to prevent disease and prevent death. So the goals are to understand frequency and distribution of diseases, and that's over time and space. So when, where does it happen, and who's affected? We want to identify disease origins or outbreak origins. We want to clarify determinants of disease. So what are those things that make some people susceptible to disease while other people remain disease-free? And these could include factors about a specific person, say their immune status or nutrition status. This can affect or be impacted by characteristics of the agent, the pathogen itself, or environmental conditions, or climate. So a lot of things can be disease determinants. And then we want to develop effective prevention and control strategies. So that's our ultimate goal, again, to prevent disease and death. So we accomplish this through surveillance. So this is routine monitoring of diseases in the world that we're interested in. We identify and describe disease occurrence, outbreak, and cluster investigation, and then through research activities. So just to give everybody a biology review, we are really concerned with the types of infectious diseases. This is something that's important in the study of infectious disease epidemiology. So of course, you have bacteria, viruses, fungi, and parasites that cause, that we consider to be communicable diseases or infectious diseases. So just to give you guys some examples of some bacteria that cause disease in humans, we have, this is Salmonella, Staphylococcus aureus. This here is Yersinia pestis, so this is what caused the plague and the culprit in the plague epidemic. This is Leptospira interrogans, a disease that causes massive morbidity throughout the tropics and one of the most widespread diseases worldwide. And we'll talk a little bit more about what zoonotic and vector-borne diseases are and such as we go on. So some disease manifestations of clinical disease, this would be cutaneous anthrax. And so so some diseases cause many disease presentations. Other disease presentations have many causes. So just to show you what some of those causes are, so this is a bacterial disease. This is a syndrome called diarrhea or gastroenteritis. So this is one of those that can be caused by many, not just bacteria, but caused by parasites, viruses as well. This is a disease called Buruli ulcer. And then these would be different presentations of the syphilis bacteria. And then pneumonias. And pneumonia is another one that can be caused by all sorts of diseases or all types of disease agents. So viruses, here's just HIV virus, influenza virus, influenza A, here's Ebola. These are some examples of diseases caused by viruses. So this is an HIV infected individual, and his clinical presentation there is Kaposi sarcoma, herpes simplex virus, dengue. So dengue causes various manifestations as well, and depending on its disease severity and the host immunity. And then a disease that you guys will never see, this is smallpox. So some infectious parasites. I particularly think parasites are interesting. How can you not think they're cute? So this is Giardia lamblia. Here's hookworm, one of the species of hookworms. That's actually his mouth, and those are choppy, bitey plates that help him attach and gnaw on the intestines. So this is a blood smear under a microscope. So this is a thin streak of blood across a microscope, and these are red blood cells. These, however, are parasites, and this is Plasmodium. So this is the parasite that causes malaria. Similarly, these are red blood cells. This is another blood parasite, and this is a parasite called Trypanosoma, and this causes Chagas disease. And then one species causes Chagas disease, and the other species causes African sleeping sickness. And then this guy is a big flatworm, and this is a Fasciola hepatica. So this is something that will attach to the liver or other organs and cause direct pathology to the tissue. So some of these, particularly this one, this is, you do not need a microscope to see this guy. He's big. So some parasitic diseases, this is Aloe, so it's a worm that affects the eye, can cause blindness. This is another worm affecting the eye. This is a disease called river blindness, and it's called by Onchocerca. So it actually causes damage to the eye. It also causes skin manifestations. So this is the worm that crawls. The larvae crawl under the surface of the skin. It causes severe itching. Individuals scratch those itches. Their skin loses elasticity, and they become physically disfigured all over their body. And this is a severe presentation of malaria. This is a child with malaria, so children are particularly susceptible to severe malaria and death due to malaria, severe fever, and sometimes even hemorrhage manifestations. So this is a worm parasite, Ascaris, and this is typically resides in the gut. This child has so many worms. So he's been exposed likely his whole life in his environment to these worms, and so they've infected him, and he has so many that they actually are filling up his gastrointestinal space, and they will come out wherever they can. And this is a parasite called leishmania, and it causes, again, disfiguring physical manifestations as it sort of attacks the mucocutaneous tissues. So not every individual will have every one of these disease presentations. They vary by the host pathogen interaction. You guys probably kind of remember this triangle where the host, the agent, and the environment act together to produce a disease manifestation in the individual. Well, similarly, we have a lot of interaction, or a large network of events that happens in infectious diseases and in transmission of infectious diseases. And so our goal in epidemiology and in preventing disease is to understand what these pieces are, and also to try to find a way, where can we intervene? So we're looking for an opportunity to disrupt this relationship and stop disease and disease spread. So it's important to think about different classifications of infectious disease and understanding what those mean. So two really important disease classifications include mode of transmission, that's how the agent or disease is spread, or how an individual acquires it, and then the reservoir, where is that found typically? So some modes of transmission are Airborne, so this is an agent that's can suspend in the air or can be carried on the air by a nuclear, so typically a speck of dust or something that would hang in the air. So indirect contact is another mode of transmission, and this is really where a pathogen has been deposited or resides on something. So think of a table or a doorknob. So this is an indirect contact, so it's been left there, and then you would touch it and pick it up and then infect yourself. Food and waterborne through ingestion, and then vector-borne. Vector-borne is delivered to you by a vector. So airborne, this is important because they're circulating on air currents. It makes them really hard to eliminate. So these can be resuspended or aerosolized by contaminated dust. So an example of this would be something like measles. So measles is airborne, and it can be coughed out in maybe a tiny droplet, but actually it can just be coughed out and suspend in the air itself. It doesn't require a droplet to hang in the air. So that'll be some droplets we'll talk about in another slide. So this is also hantavirus. So hantavirus is a virus that infects rodents. So it's a zoonotic disease. When they urinate and defecate into the dust, there's attached to dust particles. So then when this dust is disturbed, such as a farmer going out to sweep his shed, if this virus is present, it gets aerosolized, so kicked up into the air along with the dirt particles, and then the farmer who's sweeping the shed can inhale it and become infected. So indirect objects, as I mentioned, so inanimate things, things like chewing on your pen, doorknobs, tables, clothing, bedding. So anything that can pass a pathogen from an individual or an animal to another person without direct contact. Food and waterborne, again, this is ingestion, so this can be any number of things. It can be toxins, it can be direct pathogens. Sometimes it's obvious in the food and sometimes it's not. And so this is really difficult when it gets into a water supply. So there are agents such as Leptospira that I mentioned before, that little curlicue spirochete, and this is something that is also a zoonotic disease of rodents, dogs, and many animals, and they shed it through urination and defecation, and sometimes that gets into a water supply that people use to drink, so such as rivers and streams and places where those are the water source. Diseases like that are a major problem. And then vector-borne, so these are things like mosquito-borne diseases that, so think of Zika and West Nile and dengue and chikungunya and malaria, but also ticks, fleas, and I like to call toddlers vectors, so even though they're not introducing it necessarily into your body, they are bringing you all kinds of diseases. And then you often hear about fecal oral. So these are diseases that are transmitted typically person to person, but these come from, so think of something that causes a gastrointestinal illness or diarrhea. And so you might shed this out in feces and then somebody else picks it up and ingests it. This is typically through contaminated food or water. So think of Typhoid Mary, you guys may have studied in the past, where she was spreading typhoid through preparing food. Or through indirect contact. So this is also such things as doorknobs, particularly think of bathroom doorknobs, or any way that you might get some bacteria that was transmitted through feces onto your hands. And then of course we touch our hands and to our face and mouth and eyes constantly without even noticing. So direct contact, this is direct physical contact with the agent or infection. So remember I showed you that image of anthrax that looked like a black circular sore. So this is actually the bacteria coming in contact with the skin and causing direct disease there. Droplet spread, so other direct contacts, kissing, sexual intercourse. So sexual intercourse introducing things such as HIV, direct contact with body fluids, and other diseases that contact that way. And then droplet spread actually requires direct contact with aerosolized fluids. So this is important because it's different than airborne, because These viruses and bacteria actually fly on the droplet. So this is kind of a gross image of a sneeze, just to show you sort of that projectile. So larger droplets tend to fall quicker, and then smaller droplets tend to fall later. This is important in understanding disease transmission, and it may be different for each disease that we're interested in. So understanding if something is droplet or airborne is important in terms of how far it can spread and if it will hang in the air. So it's not just coughing and sneezing, but so certain hospital procedures will aerosolize this, such as placing an intubator, so somebody who's ventilated, occasionally through talking, and then through laboratory work. So reservoirs of disease, this is where the virus normally resides. So animals can be reservoirs who are not sick by the pathogen they're carrying, but also can be affected. So the environment, water, soil, I mentioned certain water supplies that could become infected, then that becomes a reservoir and it's accessible to other animals and to people who might use that. And then humans. So humans are, again, back to typhoid Mary. Typhoid Mary was a human reservoir for typhoid fever, for salmonella. Okay, so this is the category of zoonotic diseases. So where diseases that normally reside in animals, but can be transmitted to humans. Sometimes it's a rare occurrence that's transmitted to humans, and other times they're very common human diseases, but they require animals in order to sustain them in the environment and in their life cycle. So let's talk now a little bit about outbreak investigation and how, so this is a major activity in infectious diseases and is relevant right now. So I thought we would talk extra about this. So in general, this is a very simplified view of, this image is a very simplified view of how we investigate outbreaks. So those steps generally are confirm that there is an outbreak and that there's a problem that needs to be solved. Describe, so we describe what's going on and then determine the cause. Sometimes this is easier than other times, and ultimately we're going to control measures. So these include things like epidemiologic analysis and some detective work. So we'll talk about some of these. And then some of these steps will be repeated. So you may describe and determine the cause, and then go back and describe again, and then determine risk factors that ultimately help you feed into the cause. Control measures may need to be reevaluated over and over again. So these steps are in generally this order, but can be circled back to each other. So just first, let's talk about some definitions. I'm gonna hopefully give you guys some definitions during this lecture that you can use moving forward. So endemic diseases are those that occur all the time, or can occur all the time. So an endemic disease that we have in the United States might be something like E. coli disease. So it's always there, it happens sometimes. If there's an unusual event, we might consider, or we might investigate and to determine whether or not that's an outbreak, but there's sort of always cases there. This also includes things like influenza. Even though it's seasonal, we always expect it to be there. So we've come to expect seasonal peaks. But we sort of know as epidemiologists what those peaks look like, how many cases we expect to see in those seasons. And certainly if we had something out of the normal season, we would consider that a potential event to investigate. All right, so then sporadic cases. So endemic diseases may or may not be of concern. There are a lot of tropical diseases that are endemic to low income areas of the world with a tropical climate. In fact, most of the diseases we battle are there. And it doesn't mean that they're not a problem. It just means that we know that they have a baseline. Endemic diseases can easily become epidemic or outbreak or pandemic. So sporadic diseases are those that we don't see all the time, but we do expect them to pop up occasionally, and it's not a big red flag. So this would be something like a disease that we have in the United States. It might be something like plague. So if a case of plague popped up, we know that we're not expecting to see it all the time, but if a case happens, we kind of know how it happens and we might be able to track it. So we're not too worried necessarily. This depends on the disease. So if a case of smallpox popped up, we'd be extremely concerned because it's a disease that's not supposed to be here. Also, if it's a disease that's never existed before and popped up one case, then that would be enough to warrant an investigation and consider it an outbreak. So then outbreak is something that is unexpected. So an epidemic would be a large outbreak, usually affecting one region of the world. It can be a large number or a small number. Again, this is relative to the disease. Think about in terms of this, if this was smallpox, then one case is indeed an emergency. 2 cases or 3 cases is a massive public health emergency. But if this was something like pneumonia, then that's something that we kind of see all the time and can be caused by a lot of agents. It might not raise any bells at first. So that's epidemics. And then pandemic is a situation where we have several clusters, outbreaks, or epidemics occurring in various regions of the world. And then this is actually a term that's been debated a lot recently in our current pandemic situation with coronavirus, and that's because this sort of existence of the disease on many continents or regions of the world, it still needs to fit the unexpected criteria, and then there's some debate about if these traveled there and then don't stay, go away with the natural course of the disease, is that really a pandemic? So some have suggested that if a case, say, in Florida was the outbreak in Florida, and then somebody traveled here to Latin America and brought the disease, but then they recovered or they died and nobody else got it, then it might not be considered a pandemic. But if they then brought it home to Latin America and then infected their community and it started to spread again, that could be considered a pandemic. So that's a general definition, and there has been, again, it's sort of a fluid definition. So there's not like a line demarcating saying now it's a pandemic. Okay, so I've given you some of these definitions here. Cluster just refers to a cluster or a little aggregate of disease that is makes us curious. So we might look at that and call it an outbreak. We might look at it and say it's endemic. We might look at it and say it's sporadic. It just is a few cases occurring grouped together that we're interested in looking at. So it's not necessarily an outbreak, but it's something that piques our interest as epidemiologists. So how do we know if it's an outbreak? And this is just at the very basic definition, an occurrence of disease that's more than expected. So with something like seasonal influenza, we might see this seasonal peak, and that might be normal to us. But if this was something else, say measles, and so maybe there are some cases of measles that normally occur, and so this might be something that makes us, raise our eyebrow and say, well, is this what we expect or not? So in a case with this steep of a curve, we would probably say that's an unexpected number of cases, and then look at why we saw that sudden increase in cases. So just to rewind a bit, this y-axis is the number of cases, and this on the x-axis is time. So over time, we see, every day, maybe 2, 3, 4, up to 5 cases. So maybe 1 to 5 cases normal. And then all of a sudden, within the span of a week, we jump to 30 cases. And this depends on each place. So this is specific to disease. Again, if this was smallpox, then we expect to see 0, 0, 0, 0, 0 forever. And if we saw even one blip up, then that would be an alarm. So just as an example, so it's really common to have say, illness in school, and if we have students becoming sick, say with nausea, vomiting, gastroenteritis, and if we had normally expecting only one person over the course of, say, a month to develop this, and all of a sudden we had 20, we again would raise our eyebrow, call this a cluster, and think if we need to investigate it. And in this case, there are 20 times more sick students than we would expect for that same period of time for that same school. So again, this is relative to the time and place and population. So just to quiz you, is there an outbreak? We have 3,300 cases of West Nile virus infection in the United States in 2015. So we don't really know that that's whether or not that's an outbreak unless we look at a baseline number, so the expected number of cases. So we compare that to, in the previous 2 years, there were 149 cases. So do you think that that's an outbreak? And would we investigate it? Yes. In the second example, 2,820 cases of tuberculosis. So this would be in Guatemala in 2015. So as a comparison to our baseline expected data, 3,059 cases occurring in the prior year. So just given this information, because it's not above what was expected based on the prior year information, the answer is no. We would not consider that an outbreak or investigate it. Now there, of course, this is just a snapshot in time, and we would want to see, further back and look at the trends. All right, so let's talk about how we describe this information. So we use what we call descriptive epidemiology, descriptive statistics, describing when, where, and who. So for the timing, I've already shown you some graphs over time. So this is called an epidemic curve, and you've seen a lot of these on the news during the coronavirus pandemic. So this literally is a bar graph or a column graph. It is always drawn the same way. It's not the wavy line. It's not a line graph that I showed you before. It's always a bar graph. If anybody ever shows you an epidemic curve that's a line, then epidemiologists will be pulling their hair out. If you ever end up working at a health department or in a research lab, and you will make this curve. So this is incident cases. So this is new onset cases. So a case that occurred in March and is counted in this column is not also counted here. So this is 25 new cases in March. This is 125 new cases in April. This is 300 new cases in May. So in this example, this is an epidemic curve for an unknown disease. So we're just describing this is what's happening for a disease that we don't know the cause of. This could help us sort of understand what that cause might be. So it tells us a little bit about how infectious or transmissible it is, because we see that the number of cases in February to March more than quadrupled. Same thing from March to April and April to May, and then it starts to slow a little bit and then it drops off quickly. So, infectious disease experts understand sort of the nuances of disease. And this would be disease specifics. How infectious is a disease? How is it transmitted? Is it an environmental reservoir? So knowing all of those things can help you from this graph start deciding what agent it is. So as an example, so here's an epidemic of a propagated disease. So this is a person-to-person transmission. So this would be something that's passed from one person to another. It's not necessarily, this does not require an animal to transmit it. It does not require a vector to transmit it. This would be something like a respiratory illness. So something that would be droplet spread. So a droplet containing a virus that would be inhaled by somebody else or deposited on a surface. Uh-oh. Okay, sorry, my dog unplugged my computer monitor. This is a real life work from home stuff, guys. All right, so I wanted to show you this particular example of an epidemic curve because it can be transmitted, as I said, from directly person to person through the droplet that is coughed or sneezed, or aspirated in some way. And then it can also be caused through direct contact, so touching. If an individual coughs onto their hand and then touches a doorknob or touches a tabletop, and say, for example, the properties of the pathogen itself allow it to sit there and stay infectious until the next person touches it, picks it up, puts it in their nose, eyes, mouth, and then it becomes infected. So this is a transmission cycle. And then that next person is infected and has the virus, is available for transmission. So in this example, it's measles. So this is something that you might see. So what is this? This might be the expected number of cases. However, you also see you have one case